charity which promoted high-quality research and had people like David Tyrrell on the panel before he died, and very good scientists to try and strengthen the science in the area, which is, of course, what we're trying to do with our collaborative. And she was quite an amazing lady. She and her husband established the Asthma Research Campaign, which turned into Asthma Research UK. She also established, or her husband and her established Help the Aged, which is the big charity now, which has changed its name. And then she's set up this. So quite an amazing woman, really. And she had chronic fatigue syndrome herself. She was one of the founders of the National Youth Orchestra as well. She was a marvellous cellist and was in much demand for her playing, but had to give it up on account of her ME. So really quite a fascinating life, really. But we reserve this slot for innovative speakers, and I'm very pleased to have Professor George Davey Smith, who's come all the way up from Bristol to talk to us about some exciting approaches to this field. Now, George is an amazing man. He's published over 1,200 papers. He's got an H index of 142, and he's only 35 years old, so that's pretty good going. His research spans socioeconomic, epidemiology, right the way through to genetics and everything in between, including epigenetics, which connects the two together. And it really is the most extraordinary story when you read what he's achieved so far. So this is really a huge privilege to have you here, George. He is director of the MRC Integrative Epidemiology Unit. I don't know whether they call it that now, whether it's changed its name. Still there, that's good. And he was appointed to that post in 2013. So you're really an amazing man, and no better to have somebody finish our conference off than George, who's going to raise our eyebrows to the horizon, I hope, in what he's going to tell us. So thank you very much, George.
Thanks very much for the invitation to speak there. And that's my Twitter name, Mental Random, which might become, hopefully become evident during the talk. And as I said to Steve and Esther when I was asked to speak, well, I wasn't really an appropriate person because I know next to nothing about chronic fatigue syndrome. But they said that was okay because what they wanted to hear about was about approaches to studying conditions of unknown etiology and unknown successful treatments using genetic and genomic data. So that's what I'm going to do. I'm going to use examples largely not from, or almost entirely not from the chronic fatigue field, but try to say that these approaches could be hopefully successfully applied. And what I'm going to say, I'm going to just talk about the existing situation, which might reflect my ignorance, outline of omics to be covered. I'm going to talk about the central importance of germline genetics, how to improve inferences of non-genetic causes of disease using genetics, i.e. understand modifiable causes of disease and disease progression. And then I'm going to talk about the need for very large sample sizes in this field, and then briefly about epigenomics and other omics. And then we're going to conclude on why not carry on with business as usual and the need for a coordinated and collaborative approach. As I say, the current situation, I went through and looked at the studies I could find. I hope I haven't chosen any by people who are here, but these are the sorts of things you find in the literature. Pathway-focused genetic evaluation, looking at genes related to immune function and inflammation in CFS, based on 50 cases and 121 controls, for which you have literally zero. I mean, the statistical power is literally near zero for these things, guaranteeing them to be false positives. Then a convergent genomic study, this is with germline variation and transcription RNA study with 40 cases and 40 controls. Here's a methylation study of looking at methylation of the glucocorticoid receptor with 76 exposed versus 19 unexposed, because this was looking at the exposure influences. And then a DNA methylation study of associations with chronic fatigue syndrome based on 12 cases versus 12 controls, which very simple simulations demonstrate the possibility of these things demonstrating anything useful, as was the case when such study sample sizes were applied in other fields. Now, as I said, we're going to talk about various omics, but I'm going to start with the fundamental omic, if you like, which is genomics, i.e. looking at DNA sequence. And what I'm going to talk about here are studies looking at germline genetic variations, common germline genetic variations across the genome, and trying to relate those to traits and conditions. Studies which have exploded in the last few years, obviously driven by the completion of sequencing of the human genome. And now the situation in genetic studies, studies of germline genetics in any field, back until essentially 2005, was similar to the situation I would say now, from what I can see in the chronic fatigue field, which were that massively underpowered studies were done. And with the colleagues, Helen Calhoun and Paul McKeegan and I wrote a paper which didn't make us enormously popular, but in the Lancet in 2003, which pointed out that virtually every study that the Lancet published on genetic associations that Nature Genetics was publishing at the time, right the way down to these lower grade journals, virtually all of them were just simply false positives. They were not reliable, could not be replicated. This was thousands of studies were done, and then formal meta-analyses demonstrated the fact that what was being reported were just not reliable findings. And what's interesting is virtually none of those findings, the pre-2005 era, except ApoE and dementia, which has a very particularly large effect, have stood the test of time with common genetic variants. The variants that people were looking at before the genome-wide association study era have now just gone. But the situation has just been completely transformed by large groups getting together, initially driven by the funders. I mean, the Wellcome Trust was particularly important in this regard, and they said around this time, they said, we're not going to give any of you any money unless you actually get together in large groups and do sensible sized studies. And that became the Wellcome Trust Case Control Consortium, which reported in 2007 initially, with a series of genetic variants associated with diabetes, with obesity, with cardiovascular disease, etc., all of which have stood the test of time and have gone forward to the situation where there's now literally well more than 10,000 absolutely established findings that are completely replicable of genetic variation in particular parts of the genome associated with a huge number of traits. And this is a tiny sample of the GWAS diagram browser, which you can go on to the National Human Genome Research Institute. These are just looking across the chromosomes. This is a tiny section of chromosome, and each dot represents something which is now established beyond question, essentially, as being associated with the traits. I've just highlighted their systemic lupus erythematosus. I mean, there's quite a few variants now related to this, but you can click on your disease of choice, your condition of choice, your trait of choice, and the thing lights up, but not if you put in chronic fatigue, because there are no reliably established variants associated with that condition. Now, what do robust and reliable genetic associations tell us about? Well, the first thing that they can tell us about is about pathology, or by what I mean by the disease classification, what is seen to be a sensible way of classifying any condition. Because if the classifications are sensible, and you find genetic association, then it in some ways validates the way that those categorizations are being carried out. And if we predetermine categories, we can tell about possible subcategories of the condition, but these have to be predetermined, not sort of fishing exercises looking at every single possible group. After you haven't got any reliable over all of that, you then go and fish for a subgroup effect. In subgroups, this doesn't produce useful information. But probably, in my view, one of the most powerful things with establishing associations with germline genetic variation and traits or disease, it provides robust evidence of effects of putatively modifiable causal factors for disease. So although you're using genetic variation, you're using that to inform you about things which actually could be changed. This is an approach called Mendelian randomization. So I'm just going to run through a few slides as to why and how this approach came about. And the approach came about because of the unreliability of the sorts of findings we're getting in conventional observational epidemiological studies. So we're all used to opening the newspaper and you see, eating an egg a day can raise the risk of developing diabetes, and then you open the paper the next day, eat eggs to beat diabetes. 4 a week can slash risk by 40%. So what you need to do if you want to prevent diabetes, eat more or eat less eggs. Or this lovely advert from the Boston Globe. The average American lifespan has increased nearly 3 years over the last 2 decades. So it says, we've been selling vitamins at a discount since 1977. Coincidence? We don't think so. So vitaminshop.com, who might have got a peculiar interest in this area, wanted to believe that it's because you've been taking their vitamin supplements that people are living longer. And the reason they could say this is because of observational data, studies that were done with conventional approaches that asked people whether they were taking vitamin supplements, and then for how long they were taking vitamin supplements, and then, so you can't see the line here, but then followed them up and found out whether they had heart attacks or not. And what it showed was that if people were taking vitamin supplements just for 2 years or more, they had about a 40% lower risk of coronary heart disease. So that, and they did the usual things that you do in these studies. They statistically adjusted for confounding factors, cholesterol, diet, other dietary factors, weight, smoking behaviour, drinking behaviour, etc. And the result appeared reliable, and these things get reported in the newspapers. This is the New York Times. Vitamin A greatly reduces risk of heart disease, studies suggest. And around 40% of American adults at one time were taking supplements that contained vitamin E, just because this was highly promoted. You'd probably be unsurprised to know that once randomised controlled trials were done with up to 13 years of follow-up, randomising people to either vitamin E or placebo, not only was a 40% reduction in risk not seen, there was no reduction whatsoever in the risk of coronary heart disease, despite the observational evidence. And the same thing has been seen with many other vitamins, the same thing's been seen with hormone replacement therapy, and many other things which have been tested in randomised trials. And the point is that the things that go on to be tested in randomised trials are the strongest, only the things with strongest evidence would anyone spend the money carrying out randomised trials on. So these conventional approaches to try and establish factors that might cause disease do badly. And the reason for this is probably, is often because the things you're looking at are so strongly related to other factors. That taking vitamin E is related to many other factors. Also, if you get the disease or you have disease processes started, then that might influence the exposure or something which you think is the exposure, something which is called reverse causation. We looked at vitamin E levels and other factors in a big study across the UK, the Women's Heart and Health Study, and people who had higher vitamin E levels, they were less likely to come from poor childhood. People from poor childhoods have higher rates of coronary heart disease. They're less likely to be poor now in adulthood. They were much less likely to be smokers, more likely to have a glass of Chardonnay a night, more likely to exercise, more likely to be on a low-fat diet, less likely to be obese. They're more likely to be taller. Taller people have less heart disease. And they're also likely to have longer legs within their height. And longer legs are also related to lower rates of coronary heart disease. So these are the things we just measured. So of course, a vitamin E measure is associated with all these factors and many, many more factors that are related to the disease risk, those confounding factors. But sometimes this notion of how the world is, things are all associated with each other and how confounding exists is comes to mind more when you see it visually than when you do statistics. And now this was my favourite takeaway, which is takeaway in Bristol. That's my bike kebab hanging on the handlebars. And this is opposite the Prof. It's now called the Crofters' Rights Pub. And the days I used to live near there, you'd go and smoke in the back of the room with the Crofters, having your ears blasted out till 2 in the morning, drinking their lovely beer. And you'd stagger across the road to Rita's, and there'd be loads of people similar to you in the queue. And this is the menu of these fantastic spare ribs, dripping in fat. And then it says, these dishes are served on salad, if required. And over a decade or more of participant observation, I never saw a single person who required salad. But it does bring home to why people with lower vitamin C levels or vitamin E levels have more heart attacks. You realise the things that it goes with. So much for the antioxidants, or as some people would say, maybe so much for conventional observational studies. And this was a cartoon from a paper in the US, which has the news reading, it's eggs are good for you or bad for you sort of stories. And he presses the button, and these things spin round, and they say that coffee causes depression in twins. That's today's story. And then the next day you can press it round, and, red wine. Well, actually, all of these are fake, except, all of these are non-causal, except red wine does cause sexual dysfunction in men aged 25 to 40. So it's a very distant memory. So those conventional approaches have their limitations, and Mendelian randomization is developed as a way of trying to get around the confounding, all the factors being related to each other, and the reverse causation, the fact that the disease processes might influence exposure. And it does this by using germline genetic variants that proxy for the exposure you're interested in. So there are genetic variants that reliably relate to level of vitamin C, that relate to the uptake of vitamin C across the gut. And so people who carry the variants related to higher vitamin C, as a group, they have higher vitamin C than those who don't. But they don't differ from the other people in other ways. They don't differ by social class, by leg length, by smoking behaviour, drinking behaviour, other dietary factors or whatever. They just differ by that one factor. And therefore, if you use the genetic groups based on their genotype, you get groups which are like a randomised trial. One group has higher vitamin C on average, one group has lower vitamin C on average. And if you follow them up and if vitamin C was indeed protective, what you would show was that the group with the genes related to higher vitamin C had lower rates of coronary heart disease. And so the trials suggest that vitamin C is not protective, and a huge Mendelian randomisation study with 100,000 people in Copenhagen suggests the same thing. So you can use genetic variation which relates to the factor of interest as what's called an instrumental variable in the sort of econometrics literature, because it will not be related to the confounding factors and there will be no reverse causation. So this has been called Mendelian randomisation. And that GWAS catalogue, the catalogue of all those genome-wide association studies I showed you with the 10,000 traits, those include genetic variation related to health behaviours like smoking and alcohol drinking, and it includes genetic variation related to most or a huge chunk of the biomarkers you can think of that people have been interested in studying. And this method has been applied in a huge number, a very large number of fields now, and has certainly provided reliable evidence, as has been shown by situations where randomized controlled trials have tested the same things as Mendelian randomization. So this is an example of a protein called C-reactive protein, which is an acute phase reactant. If people are ill for virtually any reason, the C-reactive protein goes up, and it predicts coronary heart disease very strongly, even after you adjust for all the confounders. And pharmaceutical companies were developing drugs targeting C-reactive protein a decade or more ago. But because of the problems with observational epidemiology, these observational studies adjusting for all the confounders might well suffer from residual remaining confounding or reverse causation, like the vitamin E story. But then if you have a genetic variant which is in the gene that produces this protein, then that genetic variant will not be related to all the confounding factors, but can be used to classify groups that have, on average, higher level of CRP or not. And then if CRP were causal, you'd expect higher rates of coronary heart disease for the people who carry the variants related to higher CRP. This association. What was actually, and this allows you to predict what the causal effect of modified CRP levels would be. Because the joint effect of the genetic variant on CRP and the genetic variant on the outcome allows you to mathematically estimate the protective or non-protective effect of vitamin C, of CRP. In this situation, in this case, it was shown to be non-protective, and the development of drugs targeting this stopped. So I'm just going to show one recent study, just because it's very recent. This was a study we did looking at vitamin D and the risk of multiple sclerosis. There's been a very large number of observational studies suggesting that vitamin D might protect against development of multiple sclerosis, including the fact that basically the major identifiable factor is that the closer to the equator you lived as a child, the lower your risk of multiple sclerosis. I thought this was not going to be causal. I was very sceptical about this association. But we did the study looking at genetic variation related to vitamin D. This is just what you do when you do such a study. You look at what's known about the production of the biomarker, in this case vitamin D levels, and you look at genetic variants which have been related to any of those steps in the production of vitamin D. And these are these variants, and the vitamin D variants do indeed relate very robustly to vitamin D. If vitamin D were protective, you would expect to see that the variants would also be related to a lower risk of multiple sclerosis. And this shows p value 10 to the minus 19 is that if you put that, add the genetic variants together into an allele score, this is the declining average vitamin D levels and the ranges for the groups classified by these genetic variants. Now those groups will not differ on all the confounding factors that we've discussed in the case of conventionally measured risk factors. And what we found, as I say to my surprise, was that the genetic variants associated with higher vitamin D on different genes and working through different pathways were all associated with lower risk of multiple sclerosis. So this does seem that there really might be something in the story that vitamin D protects against the development of multiple sclerosis. And this is just because this is a story 2 days ago, and I'll finish on Mendelian randomization with this. This was Eli Lilly and drug development sending stock plummeting. And this was a trial which had been carried out on an HDL cholesterol raising agent, which Mendelian randomization studies for some time have suggested that what we've all learned of as being good cholesterol for so many years. We've heard that HDL cholesterol is good cholesterol. The genetic variants related to HDL cholesterol were not associated with any difference in risk of coronary heart disease, unlike the genetic variants related to LDL cholesterol, which very robustly relate to coronary heart disease, demonstrating the causal nature of LDL cholesterol. And no more trials will be carried out of HDL cholesterol raising drugs because so far 7 or 8 of them have been carried out at the cost of $100 million each, and all of them have failed. And the genetic studies would have predicted that. So certainly the pharmaceutical industry is very interested in this approach of using genes for sorting out or getting better evidence on causal factors. So robust and reliable genetic associations give us some evidence about factors that might be causally related to disease. You can sort out cause and effect with bidirectional Mendelian randomization, and this means if you think, that people have thought that vitamin D and body mass index are related to each other, as they are, and there had been an argument that vitamin D actually lowers body mass index. But if you do the Mendelian randomization study from vitamin D to body mass index, it suggests no effect of body mass index of vitamin D on body mass index. Vitamin D doesn't make you thinner, but if you do the Mendelian randomization from body mass index onto vitamin D, it suggests that the direction of causation runs from higher adiposity leads to lower circulating vitamin D levels, which makes sense. So you can do the Mendelian randomization in both directions, and we also did this with which are multiple sclerosis genetic variants don't relate themselves. The ones related just to multiple sclerosis don't relate to vitamin D levels. You can put it in that direction, for example. Reverse Mendelian randomization can suggest causes that haven't been thought of, i.e. if you do a genome-wide association study looking looking at genetic variation right the way across the genome, and you find that genetic variants in a particular pathway are related to the disease, then that would say it's worth looking at that pathway as being a potential causal factor. An example of a reverse Mendelian randomization study, although the cause was already known, was a series of 3 huge genome-wide association studies of lung cancer that were done at the cost of tens of millions of dollars at the time. And all 3 of them came up with one gene region, which was a nicotine receptor region, which was associated with smoking more. So if we hadn't known that smoking caused lung cancer, that genetic association study would have given us very strong indication that smoking was likely to be a risk factor for lung cancer, because you found genetic variants related to smoking were related to lung cancer. So that sort of reverse form can suggest factors that might be causes of disease that you haven't thought of, that you would want to look at further. You can use a method which I won't go into detail about, which is a method called LD regression, which is a method if you have the genome-wide association data on a huge number of traits or conditions, including the one you're interested in. You can see whether there's actually just genetic correlation between your condition or your trait and the others. And if one trait caused another, you would expect to see such genetic correlation. Now, the advantage of this, and the advantages of the Mendelian randomization studies, is that you often don't need to have the individual level data. And you don't have to go out and collect it, because there's a huge amount of genome-wide data which is available. You know, the multiple sclerosis study we didn't do a huge genome-wide study of multiple sclerosis. We used the data which had been produced, starting with the Wellcome Trust Case Control Consortium, that had been produced on multiple sclerosis, and we used those data and then looked at the genetic variants related to vitamin D in those data. And one of the fantastic things in this field is that huge amounts of such data are just available to analyse. And so you can use this vast amount of available data to strengthen what you know about the condition of interest. And this is a summary just of sort of showing what you can do with LD regression from a lovely paper in Nature Genetics, just appeared online. But it shows that with sort of no hypothesis at all, you show that BMI is related to type 2 diabetes, for example, and age of menarche, later age of menarche is related to lower BMI, those sort of things. So these are sort of fishing expeditions you can do, but using all the available data to see whether your trait appears to be linked to another trait for which genetic data are available. And then you would follow those up in formal Mendelian randomization studies. Pathway analysis, if properly done, can suggest that particular mechanisms might be involved in the condition. If you actually know a pathway like the vitamin D synthesis pathway, we can show that the variants right the way down the pathway are related to the outcome. That then sort of strengthens evidence that that pathway is involved, or you can then run to more elaborate pathways. And you can then look at what nodes in the pathways might be important. So, for example, in the CRP story, that was a disappointing story, because CRP turned out not to be causally related to coronary heart disease. But interleukin-6, which upregulates CRP, the Mendelian randomization studies suggest that that is related to coronary heart disease, and a randomized controlled trial with receptor inhibition of IL-6 is ongoing at present with coronary heart disease as an outcome, which is supported by those genetic studies. Okay, what does the use of genetic association data not tell us about? Well, at present, it tells us about nothing if the sample sizes are too small. And by too small, you mean basically less than many thousands at least for common complex diseases or conditions or traits. You need very large sample sizes to get reliable evidence. And in fact, worse than having no data at all is actually having inadequate sample sizes, which mislead people, which further research is carried on about, and funds wasted in that way. So it tells you nothing if you don't have adequate sample sizes. In the present situation, gene-by-environment interactions, mostly at present, and nearly all of them don't replicate. The gene-environment interaction story is rather like the candidate gene story was like in 2003. when it's producing findings that don't replicate. Probably just because so many potential interactions can be analysed, so much fishing can be carried out, and replication has not been the norm. And it doesn't, and very importantly, I think, it doesn't necessarily tell us about treatment. If your genome-wide association study data are about actually having the condition or not, like having lung cancer or not, a Mendelian randomization study will tell you about factors that are related to developing the disease. Now, the genome, Mendelian randomization says that smoking causes lung cancer, which it does. But once you've developed lung cancer, stopping smoking is not an effective treatment. And that is the case with many diseases. And I think it's something that's sometimes inadequately realised, because when genome-wide association studies of diseases were established, there were many claims about this is going to tell us about cures. It doesn't tell you about cures. Genome-wide association studies of having a disease or not tell you about factors related to developing the disease. And in many conditions, once the disease has been triggered, then intervening in what might have triggered the disease no longer has any effect. As we obviously know, it doesn't in the case of smoking and lung cancer. And that's likely to be the norm. We've probably been somewhat misled by the fact that coronary heart disease, for which so much epidemiology and clinical work has been based on, in coronary heart disease, factors that increase its risk, like cholesterol, blood pressure, and smoking, if you intervene in those, once people have had a heart attack, it also reduces having a repeat heart attack, which is what you're trying to reduce. But treatment of conditions that have been established, like cancers and maybe some immunological conditions, once the disease has been triggered, then targeting the trigger no longer has any effect. So for any condition, you need two studies. You need a genome-wide association study of people having the disease versus not having the disease. And then you need a really well established cohort of people with the disease. And then you look at genetic variants related to progression of disease. And there's been really remarkably little work in any disease area looking at common genetic variation related to progression. But things related to progression, once you've got the disease, they would be factors that would tell you about potential treatments. So two studies, two basic study designs are required to get the best information about disease prevention and disease progression and resolution. Okay, so I'm going to be ludicrously brief on the other omics, so I don't run on too long beyond my welcome. I'm going to talk. So when you collect a blood sample, you can look at epigenetic markers on the genome, proteins, metabolites, and antibodies, and other things. So I'm going to talk a little bit about epigenetics, as this is a very fashionable area. So epigenetic markers, they're on your germline DNA, but they basically influence whether that DNA is actually expressed, whether it's actually sort of switched on or not in a cartoon form. And so there's been lots of interest in epigenetics because it might tell us how the environment influences or works together with the genome to develop disease. And this is the idea that exposure might influence epigenetic processes. The epigenetic processes might mediate the effect on disease. But the problem with the epigenetic measures is that they are phenotypic. They're like vitamin E and C-reactive protein, etc. They are not protected like the germline genome is from change. They change all the time. That's the whole point of them. And so they will be related to confounding factors. Reverse cause will be at play. Disease will influence your epigenetic processes, etc. So they don't have the same privileged status, and nor does gene expression in a similar position. They lie before gene expression. And of the various epigenetic processes, DNA methylation, which is probably the more stable, longer-term effect, but also is the one that can be measured. So technically, it's the one that has been most studied because it's something that actually can be measured on large population sizes. As I say, epigenetics is the current genomics of choice. So the confused researcher's friend. If they ask you anything you don't know, you just say it's due to epigenetics, and people nod and say, "What a very clever person you are," when you say that. And there's definitely sort of the hyped field at the moment. This is, you might have seen these stories. DNA test reveals if you're gay. Genetic code clue is 70% accurate, claim scientists. This is a DNA methylation This was DNA methylation being related to sexuality and sexual orientation in 37 people. As a friend of mine tweeted, most mornings when I wake up, I can't be 70% sure what my sexuality is. So maybe I've got to go get my DNA methylation test. The study of Holocaust survivors finds trauma passed on to children's genes. This got a lot of attention. And this paper was 22 exposed versus 9 not exposed. So nonsense is getting published in this field as well. Now, what is it? What's new and what's true? Well, so in some ways, thinking about epigenetics is thinking about things which you can almost say are tautologous. It's very clear that processes occur which lead to the same genome being expressed differently. Because you started out as one cell with one genome, and all your cells, essentially, you've got a few somatic mutations, but your cells basically are all the same. We all have the same genome, and yet some of you developed into liver, some of you developed into brain, etc. So those processes, which during cell division leads to some cells becoming specified, and as liver cells divide, they produce more liver cells, not brain cells. Those processes are, by definition, they're epigenetic processes. So it's very clear that epigenetic processes are absolutely key to development. And in disease, the mitotic transmission of transformed cellular characteristics must be key. So this means just during normal cell division, then once you've got a diseased liver cell, if the liver is going to remain diseased, as those cells divide, they must pass on whatever it is that leads to the disease pathology. It must be passed on at cell division, otherwise the disease would be cleared. Mitotic transformation, mitotic transmission of these changes must exist. So it's likely to be involved in disease processes, epigenetic processes. Doesn't mean it's methylation, there's lots of other epigenetic processes, but some processes will be involved. And we have ways of assaying and quantifying these processes, in particular methylation. It might give us ways of studying how gene and the environment interplay. Definitely provides robust evidence of the experience and timing of some exposures. For example, in a study that was done in the ALSPAC study, a birth cohort that Gene Golding set up, where we had data on whether the mother smoked during pregnancy. We had blood taken from the umbilical cord of the baby when they're born. You could see an incredibly reliable signature of maternal smoking on the cord blood, and you could see, for some of those factors, you could see it at age 17. So you could take a blood sample, and you could say the mother smokes. It would be incredibly useful for seeing what exposures people had received, and also suggests that things might be transmitted long term. My mother is now dead a few years, but when I started working and I was looking at maternal smoking many years ago and some outcomes, especially childhood IQ and growth, and showed maternal smoking, which I suggested was confounding, wasn't of course a fact. It was a way to get a lower IQ and shorter height. She said I should sue her, so because she smoked 3 packs a day during when she was pregnant with me. Medical legal aspects of these and one of the most remarkable things, but absolutely replicates, is these methylation markers can really closely tell people's age. If you find a blood spot, you could say within 2 or 3 years this person is 40, which is utterly remarkable. But it's true, it completely replicates. So that has forensic uses. You find a blood spot at a crime scene, you say this is a 55-year-old heavy drinker who used to be a smoker. Well, nearly all crime committed by 55-year-old heavy drinkers. They used to be smokers, but this could have forensic value, and can also help in reverse mechanisms of disease. For those of you interested, there's a special issue of the International Journal of Epidemiology with 250 pages of exciting page-turning epigenetic epidemiology. Just came out a week or 2 ago, and there's an editorial that Caroline Milton wrote on stem cells to the law courts, on the forensic aspects of the epigenome and the importance of actually using epigenetic measures as what we call the biosocial archive, something which gives evidence of the sort of history of exposures that people have had. Now, stem cells are not really under the omic category, but are another incredibly interesting area of technical development. And the relationship with epigenetics is that if these epigenetic markers, like smoking or maternal smoking, if they are stable, you can see them 17 years later, then the white cells are dividing all the time, and they're being replenished. So there must be some changes which have actually occurred in stem cells to allow these things, that allows that continuity. And some of the markers resolve quite quickly when you stop smoking, or once you're born. Some of the ones that relate to smoking, resolve. Some of them just stay. So there's stem cell biology is involved. And one of the exciting things about about the stem cell situation is that if you have large banks of cells that you can reprogram to stem cells and then program forward to resemble other tissue types, you can select them once you've got genetic evidence about what genes relate to disease. You can select them and do disease modelling with stem cells. So any large population sample, I think it's worth establishing lymphoblastoma cell lines which can be reprogrammed, and the virus which is used to immortalize them washed out and reprogrammed to other forms of cells. So a very exciting area, I think, and links the epigenetics with cellular function. Gene expression is transient and was the sort of overhyped omic before epigenomics came along, and quite disappointing. Some disappointing areas where it was thought that you could have this wonderful predictor of disease progression or whatever that a lot of studies that couldn't replicate. But I'm mentioning gene expression just to emphasise again the incredible resources that are available, these huge resources that are available. The genome-wide association studies, there's huge epigenomic resources, the roadmap and ENCODE, which have epigenetic data, and there's this extraordinary study, GTEx, set up in the US, which takes autopsy material from people who've usually died suddenly, and is generating whole genome-wide expression data on the tissues. It's generating genomic sequence data, it's generating methylation data, etc., and it's open access. So, if you're interested in some of these areas, then the idea of setting up your own small study makes no sense at all. But if you have genetic variants which are definitely related to disease, you can go and look at them in these tissues to get evidence, in these data to get evidence of biological processes that might be involved. Absolutely extraordinary resources. And other omics, proteomics, the study of proteins, as the name suggests, which would be very exciting when scaled up, when made possible to do at scale. Currently, very, very, very expensive process. Metabolomics are small metabolite molecules for which there are some quite affordable platforms which generate data on some of these metabolites, but not all the 10,000 or more human metabolites. But the exciting thing about proteins or metabolites is many of them are straightforwardly druggable, i.e., for many of them it would be known how you would alter their level if they do appear to be involved causally in disease. And for all of these, you can do, not all of them, for many of these, you can do Mendelian randomization with reliable genetic variants related to them to see how that relates to disease. There's what I'm just calling immunologics, which is platforms like Somatologic, which has about over 1,000 entities, many of which are immune function related. Is expensive, but you can generate this mass data on small samples, and also, of course, obtain evidence of infection with the serology. And again, I think fighting with epigenomics, the most fashionable omics is microbiomics, or the study of the microbiome, or the 9 out of 10 cells that each of you have, which isn't you, i.e. some bug. The study of the microbiome is very fashionable and is becoming tractable in ways it wasn't. So that was a very brief run through why not business as usual, and then the need for a collaborative approach. But as I say, since I know next to nothing about chronic fatigue syndrome, I think this is an issue for people here to discuss whether it should be business as usual, whether the possibilities thrown up by these new technologies and by these amazing data resources which have been established at the cost of a small country economy, whether utilising those to the optimal in any area you're particularly interested in might be the best way ahead. So I'm not going to say any answers here. I think it's a discussion period, so maybe this will be a discussion that Steve would lead. And I'd like to thank people for listening, or for at least being discreet if you weren't listening. And I'll put it back on out loud again.
Thank you, George.